All right. So the notes start off with these things called characteristics of life, and what does that what that means is what makes living things different from non-living things, right? So when you think of non-living things, I'm thinking about, like, for example, my car. My car is a non-living thing, but my car does show some examples of characteristics of life, right? For example, is my car made up of a high degree of organization? Yeah, the car is highly organized. There's highly organized components, computerized components, right? Does it use energy? Yeah, it uses energy. We actually have to put um, a fuel source into the car, and then it uses that fuel source to produce energy, just like the body. However, it does not do many of the things uh, that living things can. It does not possess all of the characteristics of life. For example, it cannot maintain homeostasis. It can sense the environment. In fact, some cars can drive themselves and respond to the environment. But it's not going to reproduce. It doesn't contain materials only found in living organisms. So as we go over these, it'll become clear that living things, right, possess all of the life characteristics, all of them. If we have all of them, then it's a living thing. If we don't have all of them, then we can't really classify um, whatever it is, the thing that we're looking at as being living. So having a high degree of organization, that's kind of important. In fact, these two kind of go together, these first two. When we look at the human body, there's a high degree of organization, right? Well, if we look at the body and go really, really small, we can see that there's complex molecules, right, that are only found in living things. Molecules like DNA, like lipids, proteins, carbohydrates, right? But when we put molecules together, like lipids and carbohydrates and proteins, we could build cell parts and cells, and then we can combine cells to form tissues, and then from there, organs, and then organ systems, and then organisms, right? But not all living things contain tissues, organs, and organ systems. Some living things are just found at this level, at the cellular level, things like bacteria, protists, stuff like that. But even at that level, there still is a high degree of organization. All right. Even the simplest life forms exhibit a high degree of organization because they're still made up of really complex molecules. Acquiring and using energy, this is a combination of two characteristics. The first one is nutrition. All living things have to have some kind of nutrition. When we look at plants make their own food, so they show autotrophic nutrition. They use photosynthesis. So in other words, they take carbon dioxide gas from the atmosphere. They take water from the ground, and combined with sunlight, they can make their own food in the form of glucose. Animals, we show heterotrophic nutrition. So that means that we have to go out and find food. We could eat the plants, right? Or we eat other animals that ate the plants. So we are getting the glucose that was made from the plants, either directly by eating them or indirectly by eating something else that ate them. But getting food into the body, that's nutrition. And then using that food along with oxygen to produce energy, this is called respiration, right? This is also one of the unique characteristics of life. So nutrition, respiration, this is described as just acquiring energy and using energy. This one is a big one, maintaining homeostasis. What is maintaining homeostasis? It's kind of like a balance, right? If you can imagine like a seesaw. Um, Maintaining homeostasis is an ability to keep things inside the body stable when the conditions outside the body are very unstable or dynamic, right? So in the textbook, it gives the example that they give of maintaining your body temperature. All right. So when it's hot outside and you're standing out in the sun, your body temperature will rise. So your body, because you're a living thing, you have the ability to maintain homeostasis. So the body's going to generate a response to that. So what's going to happen is as your body's temperature is rising, the brain is going to receive that information. And the brain is going to generate a response. So what's going to happen is your blood vessels will become dilated, right? And that's going to allow the warm blood to get close to the surface of the skin. Your skin might get flushed, red. You're going to start to sweat. And as the sweat evaporates off the surface of your skin, it's going to remove some of the heat that's in the blood. And hopefully that'll cool you down. But also, you're going to think to yourself, oh my goodness, I'm so hot. And you're probably going to move into the shade. So some of the responses are voluntary, like you're going to take off your sweatshirt or whatever, or stand in the shade or go inside even. That's a voluntary response. But some responses are involuntary. So sweating, and stuff like that. So regulating body temperatures is just one example of maintaining homeostasis. I think another example that a lot of the textbooks use is controlling your blood sugar. When, let's say it's Halloween and you take all your candy home and eat all the candy in one night, while your blood sugar is really going to spike. So the control center for homeostasis is called the hypothalamus, and that's in your brain. The hypothalamus is going to receive the information that your blood sugar is high, and it's going to generate a response. And the response is it's going to communicate with the pancreas and tell the pancreas to release insulin, right? So as blood sugar increases, insulin production from the pancreas is going to increase, and that insulin is going to end up in your blood. And what that's going to do is going to cause the body to absorb glucose. So it's going to cause the liver to absorb glucose. It's going to cause the muscles to absorb glucose. And what that does is it takes the sugar that's in the blood and gets it out of the blood. And as a result, blood sugar goes back down. And when blood sugar is down to a normal level, then the insulin production from the pancreas will stop. All right, so this is another example, and there's many examples of homeostasis. Sensing the environment. So this is the ability to detect stimuli, right? So this is having some level of sensitivity. We have senses, right? We could touch, we could see, we could smell. So this is how we sense the environment. And why that is important, because that helps us to respond to the environment, right? A response is another characteristic of life, right? So this is all about what's called regulation. Adapting to the environment is important. Altering the environment. Now, this one is really important because this is what sets humans apart from other species, is our ability to change the environment. There's no other species on the planet that could alter the environment like humans can. And if you don't believe me, you could just go down and walk around New York City, right? I mean, what other species could build something like that, right? And then, of course, reproduction. Without reproduction, the species would be over, right? So it's an important life process to be able to reproduce. All right, so we got reproduction, we got regulation, we got respiration. We could throw growth in there. Growth is another important characteristic of life. We got nutrition over here, we talked about. Excretion is another one. It's not on this list, but this excretion refers to the body's ability to eliminate wastes, right? The body produces a lot of wastes produced by the cells. I'm not talking about food waste. I'm not talking about the undigested food that we eat. That exits the body as feces. We're not talking about that. We're talking about waste products that are produced by your cells, like nitrogen molecules and carbon dioxide, for example. They have to be eliminated from the body. So this is the job of the kidneys. They'll collect waste products from the blood and eliminate them from the body in the form of urine. Another one that it really isn't covered on this slide is transport. Transport is the ability to move substances around inside the body. It's an important part. Plants use it. Plants are able to pull water from the roots and transport that water all the way up to the tallest leaves, right? And when glucose is made in the leaves, the plants have the ability to transport that glucose all the way down into the roots. We have a circulatory system and a heart that pumps blood, and this is how we move things around our body. And then, of course, synthesis, right? Synthesis, you probably heard the word synthesis in photosynthesis. And synthesis just means to build. The body has the ability to build stuff, right? And we build molecules. Some of the things that the body is building, like we have to build things in order to grow, right? So we're building many cells every single day. So in order to build things like cells, we have to build proteins, we have to put together lipids. In fact, when the cells are building proteins, that's one of the most important jobs of cells. We call that protein synthesis. Photosynthesis, what plants do, photosynthesis literally means building synthesis from light. Photo refers to photons. Photons are beams of light. So photosynthesis, literal translation, is building from light. And of course, plants build food from light, right? So having a high degree of organization, like I said, all living things are composed of cells. But even though the smallest unit of life is the cell, cells are made of things that are even smaller than that, right? In fact, in order to make cells, you have to combine organelles, right? Organelles are cell parts. So we have to put together mitochondria, endoplasmic reticulum, nucleus, cell membrane. We throw some cytoplasm in there, right? Ribosomes. All these things are organelles. And in order to make these things, we have to combine organic molecules, proteins, lipids, carbohydrates, DNA, nucleic acids. And in order to make molecules, we have to combine atoms. All right. However, when we combine atoms, things like carbon, oxygen, hydrogen, nitrogen, to form molecules, and when we combine these molecules to form organelles, and then finally, when we combine all the organelles to form cells, it's only at this level, the cellular level, that we could call that thing living. All right. A muscle cell is a living thing, but also a bacteria, which is only one cell, is a living thing. A nucleus by itself is not a living thing. Mitochondria by itself is not a living thing. When we combine similar cells performing similar functions? Well, we have tissues. So when we combine different muscle cells together, we could create muscle tissue. And when we combine muscle tissues, similar tissues together, we can produce an organ. The heart is an organ that's made up of muscle tissue. This muscle tissue is made up of muscle cells, right? And then when we put together organs that are similar, that have a similar function, so the heart, along with arteries, capillaries, and veins, we produce what's called organ systems. In this case, the cardiovascular system. And then when we group all the body systems together, the cardiovascular system with a nervous system and a digestive system and a respiratory system and so on and so forth, we have an actual individual, a human, or what we call an organism. All right, so living organisms have a high degree of organization. So an organism is a highly complex and organized unit of life, right? A multicellular organism. But so are living things at this level, because molecules could also be pretty complex. Proteins are highly complex polymers, right? So they are made up of repeating units, but they come in really dynamic shapes and sizes, and they perform dynamic functions. All right, so if we have a visual of this, well, we combine atoms, right, to produce molecules, and here we're combining these atoms. This looks like DNA, right? DNA is only one type of molecule, organic molecule, that's found in cells. Proteins would be a big part of a muscle cell here, but also lipids and carbohydrates. And then we combine cells to produce tissues. We combine tissues to produce organs. Similar organs create organ systems, and then all the organ systems produce an organism. Homeostasis, remember, homeostasis is one of the life characteristics, right? Homeo mean unchanging, stasis means standing. So this is homeostasis is an ability. A rock cannot maintain homeostasis because the rock has no ability to do so. Living things have the ability to maintain stability inside the body, inside their cells, right? All right. So what the body's internal environment remains relatively constant, right? So we have limits that the body considers acceptable. There's a limit on your body's temperature, for example, that is acceptable. Once the body temperature increases above that limit, you'll begin to sweat. Below the limit, you'll begin to shiver. Once the glucose levels go above the limit, you'll produce insulin. Below the limit, your body will produce glucagon. All right, so again, we have conscious responses. When you get hot, you take your sweater off. And then unconscious responses, involuntary responses. These are happening internally, right? So changes in blood vessel diameter, changes in the secretion of hormones, whatever it is, depending on the condition that we're talking about. Now, how do we maintain homeostasis? Well, most of the time we use what are called negative feedback systems. Negative feedback systems. These are the most common. The body will sometimes use positive feedback systems too. these are rare. All right. So an example of a positive feedback system would be during childbirth, for example. But let's talk about that one last. The negative feedback systems are kind of what they sound like they are. The brain is getting negative feedback constantly, right? For example, let's just stick to the same example that we are talking about. As you're sitting outside in the hot sun, there are temperature sensitive receptors inside the body, in your skin, and even deep to the body that are detecting that your temperature is rising. They're going to send this negative feedback, you're getting too hot, to the control center, which is your brain, right? So the brain is getting negative feedback on a regular basis. You're too hot, you're too hot, you're too hot. So then what happens is the brain will generate a response and formulate a response, and it will send it out. Usually when it sends it out, it will arrive at what's called an effector. In this case, your sweat glands would be an appropriate receptor. It's going to receive a signal from the brain, and then the sweat glands will release sweat. The blood vessels could be another effector. They'll become dilated, allowing the warm blood to go closer to the surface of the skin so the heat can escape. So there's receptors that are detecting changes and sending signals to the brain. The brain is the control center. And then the control center develops a response, sends the response out to an effector. This is called, and then what's going to happen is the condition is going to be reversed or reduced. Hopefully you'll feel cooler as a result. This is how most of the homeostasis type control systems of the body work, using negative feedback. Now with positive feedback, like for example, the example that I gave earlier is with childbirth. When the baby is about to be born, what's happening is the head is putting pressure on the cervix. So the cervix has pressure sensitive receptors that will send signals to the brain. And the brain will get this information. Oh, there's pressure being put on the cervix. So instead of reversing that, right, the brain will amplify that because we want the baby to come out. Where in the other cases, we're always trying to reverse the negative message, right? You're too hot. We're going to reverse it. We're going to try to cool down. You're too cold. We're going to try to reverse it. We're going to try to warm the body up. But in this case, with pressure being put on the cervix, the response is going to be to send contractions to the uterus, which puts even more pressure on the cervix, right? So then that signal goes back to the brain. Oh, there's even more pressure. All right, let's enhance it even more, right? And that's going to continue until the baby comes out. So that's an example of a positive feedback system, but these are pretty rare. Most of the feedback systems designed to operate and maintain homeostasis are negative feedback systems. I really like this flow chart because it kind of covers everything pretty nicely. So again, if we go through and we start with some type of stimulus is disrupting homeostasis. Let's say you're getting hot, right? Well, you have receptors in the skin and in the blood vessels that monitor this. So we have temperature sensitive receptors that are scattered throughout the body that are picking up the fact that your body temperature is increasing. And what they are going to do, the receptors, they're going to send a signal to the control center. The control center is in the brain, right? And the control center for homeostasis is called the hypothalamus. And the hypothalamus is going to generate a response, and it's going to send this output, this response, to the appropriate place. Any area that is receiving the output from the control center is going to be an effector. Okay. So the effector is going to carry out this response from the control center and it's going to be responsible for that change. In this case, the effector is probably going to be the sweat gland. And then it's going to really start to release sweat. And what happens is, as sweat is released, there will hopefully be a return to homeostasis, right? So temperature hopefully is going to be restored. And that's going to turn everything off. The response in the negative feedback loop will decrease. But if you get hot again, then the whole thing is going to start all over again, right? So this is pretty good. As we go through the notes in the book, as we go through the chapters and we talk about all the different body systems, we're going to see these feedback loops, right? But they're going to be specific for each body system. So this is a chart that you will see repeatedly, and you're probably going to become comfortable with it. All right. So let's talk about classification. Classification is just how we organize things, right? Classification is not really a science. It's just like kind of like something that we do. You classify your dresser drawers, probably putting your shirts in one drawer and your pants and your socks, etc. You might classify nobody really has CDs anymore, but your CD collection or your records a certain way. So classification is kind of important because it just creates an environment that makes it easier to study, right? When we classify living things, we create a science of it. All right, that science is called taxonomy. All right, so what does that mean? Well, when we classify organisms, what we're doing is we're using a system of names. These names are going to be used to not only identify organisms, but show how they relate to each other. And when we classify organisms, we classify them into groups based on characteristics. So we're talking about large groups like kingdoms. And then the kingdoms, of course, are going to be like the animal kingdom, the plant kingdom, the fungi kingdom, so on and so forth. But then from there, we group them into smaller groups and smaller and smaller and smaller groups until we are left with a genus, which is a group of organisms that are very similar to each other. And then finally, a species, which is one type of organism that is able to mate with each other and produce viable young, viable offspring. That's what a species is. For example, a genus might have very similar organisms in it. A horse and a donkey are in the same genus, but however, they're not in the same species. Horses are in the same species with each other, and donkeys are in the same species with each other. And why is that? Because when they reproduce, they produce viable young, right? When a horse mates with a donkey, it can produce an offspring in the form of a mule, but the mule cannot produce its own offspring. So therefore, by definition, they have to be separated into different species. But they are close enough that they can be grouped in the same genus, right? So with taxonomy, what we do is we take these genus and species names and we create a scientific name. Every living organism has a scientific name. The scientific name actually has 7 parts. because it has a kingdom name, a phylum name, a class name, an order name, a family, a genus, and a species. However, this gets a little bit too complicated. So what we've done is we only use the last two. All right. Um, why has the scientific community done this? Well, because in the scientific community, we have scientists that are all over the world and that refer to organisms as different things, right? So there's a lot of different common names that are used for specific species, right? Like if we're talking about like a wild cat, are we talking about a puma? Are we talking about a cougar, a lynx? There's so many different regional names of organisms that sometimes it becomes confusing. But when we assign a scientific name, there's only that one name, and it avoids confusion among scientists. So it's kind of it's an important um, thing to classify living things and use taxonomy, and specifically use this idea of giving every living thing a genus and species name. When we use these two names, this is called binomial nomenclature. Binomial means to name. Nomenclature means naming system. So we use a two-name naming system that assigns every living thing with a genus and species name. All right, so every species is given a name, right? Like the when we talk about human beings, for example, the scientific name, if we go back to this, would be well, it's Homo sapien, right? So the genus name is Homo, and the species name is Sapiens. But the family name is Hominidae. Um, the order is what's the order name? I kind of forget it already. The order name is Mammalia. Or no, the class is Mammalia. So it's like animal kingdom, chordate, vertebrata, mammal, hominidae, Homo sapiens. So that's the entire scientific name, but it gets too complex. So we just cut off, the big chunk in the beginning, and we refer to the last two names. All right. So this is an organism here. This is like an ecological hierarchy that goes beyond the individual. So if we look at the individual, we refer to that as an organism. When we group individuals in a given area, we refer to that as a population. So all of the individuals of the same species that are inhabiting a very specific area, we refer to that as a population. When we combine populations, right? For example, there's a human here, and an elk over here, and some trees in the back. We would refer to this as a community. When we combine the communities, all the populations, and then we also include the abiotic factors, right? Or the environment, we call this an ecosystem. So we have trees and grass, those are living things. People, there's probably squirrels and chipmunks and deer down here. But when we talk about the water and the rocks and how the living things are interacting with the gases down there, the oxygen and whatnot, so we refer to that as an ecosystem. And on the planet, wherever we find ecosystems, we refer to this as the biosphere. So anywhere on Earth that we have ecosystems, is the biosphere. Well, let's get back to classifying living things. Let's get back to taxonomy. Um, well, the first thing that we do is we organize things into domains. Um, the first two domains, eubacteria and archaebacteria, only include bacteria. The reason why they get the two domains is because eubacteria are very common, where archaebacteria are very rare. So rare that the scientists decided they need to be put into a different group. These guys are rare because they live in really extreme environments. All right. So we could find some bacteria that are found deep in volcanoes that could withstand extreme temperatures. We find some, and we refer to those as thermophiles, extreme thermophiles. Or we see some bacteria that live in really salty conditions that where no other things can possibly live. And we would refer to those as halophiles. So it is believed that the conditions on Earth billions of years ago were really extreme. And the fact that these bacteria can survive similar conditions leads scientists to believe that these were the first forms of life to appear on Earth. For that reason, they get their own group. Now, common bacteria are the bacteria that we find in our fingernails and on the surface of, the desk and whatnot. The bacteria that sometimes get into our bodies and make us sick. We refer to those as eubacteria. These are common bacteria. Everything else falls under the domain eukarya. So what's the difference between eukarya and the bacteria ones? Well, the bacteria ones could also be referred to as prokarya. And the difference is they have no nucleus in their cells, these guys. Where the eukarya, they do have a nucleus in the cells. In addition to a nucleus, they also have membrane-bound organelles, right? So they have mitochondria, they have endoplasmic reticulum, stuff like that. All right, so archaebacteria, the rare bacteria, the common bacteria, and then we have the eukarya. Now, the eukarya, we can break down into these different kingdoms, right? So bacteria is one kingdom. But then we have protists, fungi, plants, and animals. Well, what's the difference between what sets apart protists from animals, fungi, and plants? Well, protists have a nucleus. So that's what makes them different from the bacteria. But what makes them different from animals, fungus, and plants? Well, they are single-celled. Single cell, but they do have a nucleus. So that puts them in their own group. Now, what sets animals apart and fungus apart from each other? Well, animals are heterotrophs, so they have to find their own food. They're multicellular. Fungus are plant-like in the sense that they have a cell wall. However, the cell wall is not made of the same substance that plant cell walls are made of. Plant cell walls are made up of cellulose, which is a hard carbohydrate. The fungi cell walls are made up of a different carbohydrate called chitin. Chitin is a molecule that's also found in like exoskeletons of crabs and insects and whatnot. But their form of nutrition is also very different. Plants can make their own food from the sunlight. Fungus cannot. So they actually feed on dead and decaying matter. And then what they do is they return some of those nutrients back into the soil. So these guys are very important in the ecosystem because they play the role of decomposer. Plants are autotrophs, they make their own food, and animals are heterotrophs. So they go find food, eat it, and they use all the nutrients for themselves. They're more selfish like that. Where decomposers, they're doing that. They're eating and using some of the energy, but they do return a lot of the nutrients back into the soil. So they're important in the process of decay, breaking things down. All right, so we have this hierarchy of taxonomy. You could come up with some type of mnemonic device here. Kings play chess on fiberglass stools, or King Philip came over for good spaghetti, or King Philip came over for good soup. And that will help you to remember the levels of organization. But just keep in mind, what's important here is we take every living thing and we place it in each of these categories. And then at the very end, we use these last two to assign a scientific name. Again, the scientific names are used through the scientific community because they avoid confusion. So when we're looking at how humans are organized using taxonomy, we're first placed in the animal kingdom. Where chordates were placed in the chordata phylum, meaning that we have a nerve cord that extends down the middle of our back from our brain, right? There's a subphylum called vertebrata, meaning that we have bone that protects that. We're in the mammal class. That means that we provide milk to our young through mammary glands. We're in the primate order. We're in the Hominidae family. So that means that we walk primarily on 2 feet for moving around. We have large brain cases compared to the rest of the sides of our bodies. So that puts us in a group of individuals that are similar. So Homo. And then the species would be sapien, the only living organisms in our species with a unique set of combined characteristics from our family: bipedal, opposable thumbs, large brain case. All right. And of course, we would use these two. And sapiens isn't written here. All right. Those last two would give us the scientific name. All right. What do we have here? Scientific method. What is the scientific method about? Well, the scientific method is a series of steps, right? That's all it is steps. If you want to perform an experiment, you got to follow certain steps. These steps are called the steps of the scientific method. And there's certain rules, right, that come along with using these steps. So pretty much we break it down into these different steps here. The first one would be an observation. Let's say, for example, the observation it could be anything like my students are sleeping in class, right? This is also a problem. Sometimes observations, it won't say observation, it'll say a problem. So what would you want to do next? Well, you would probably want to gather as much information as you possibly could at this point. So what does that mean? So that could mean like maybe consulting with other professionals. teachers, hey, I'm noticing my students are all falling asleep. What do you do to keep your students awake? Or you might want to go on the computer and Google it or go to the library or something like that. You want to gather as much information as you can. So let's say you talk to somebody, one of your colleagues, and they're like, oh, you know what I do? What I do is I put up a coffee bar. I have a coffee bar, and when the students come in, they can help themselves to coffee and keeps them awake. So let's say you want to try this. You want to experiment with this. So then you would develop a hypothesis. You would predict. A hypothesis is a prediction about how the results are going to end. And so what you would do is you would use what's called deductive reasoning. And what that means is you would create like an if-then statement. So then you would say, if I give my students Red Bull when they walk in, then they will stay awake. So this is the prediction that you're going to make. So an important part of the hypothesis is actually saying the prediction over here. So if X, I give my students Red Bull, then Y. This is what's going to happen. You can't say then maybe Y or then possibly Y. You have to say why this will happen. The 90% of the students will be awake. 95, 100%, whatever it is, you have to say. You have to predict. And then you're going to actually go out and perform this experiment, right? So you're going to run this experiment. You're going to gather all the materials together. You're going to write down the methods, what you're going to do. And then you're going to collect the data. Not only when you collect the data, you're counting the number of students. You're going to organize this data, put it into charts, maybe make a graph out of it. When we organize the data, it makes it easy to analyze, right? So when we do all that, we could then come up with a conclusion. When we're coming up with a conclusion, what are we doing? Well, we're going back to the hypothesis and the data, right? We're answering this question: does the data support my hypothesis? It's pretty much all you're doing when you're making a conclusion, is answering this question. Does the data, right, support my hypothesis? So you're going back to these two things. Does this say what I predicted it was going to say? And if it does, then you want to repeat that experiment. Do it again and again and again, and have other people repeat it, right? So you want to communicate the results, sharing the results. And when the experiment is repeated many, many times by many, many different groups of people using your methods, your exact methods and your materials, well, then it'll gain some traction, right? But if it does not, if the data does not support the hypothesis, if you're giving Red Bulls to your students, and I'm having iced coffee right now, I'm staying awake pretty good. If you're giving a Red Bull to the students and they're still falling asleep, then the data does not support the hypothesis. So in that case, you'd have to go back to the hypothesis and modify the hypothesis. So these are the basic steps that we would use in performing an experiment. Now, this example uses this idea of the rooster crowing in the morning, right? So the observation is you're observing that every morning this rooster is crowing at dawn. So you're predicting that the rooster crowing is causing the sun to rise. That's your prediction. You're predicting that this rooster making this noise is what causes the sun to rise. Obviously, this is nonsense, right? So in the experiment, if you want to test this hypothesis, what you would have to do is you would have to kill the rooster and wait and see if the sun will still rise. So with the alive rooster, you would see that the sun is, you can't see that there is a bar here. It's just colored differently, right? You would see that out of 10 days, the sun still rose 10 days. And even with the dead rooster, the sun rose the same amount of days. So when you're coming up with your conclusion, does this data support my hypothesis? No, this data does not support my hypothesis. I predicted that the rooster was causing the sun to come up, but I could see even if the rooster's dead, that the sun is still coming up. So this data does not support my hypothesis. So then you would communicate that to their community. The sun rises even if the rooster is dead. All right. So there are these things called variables that are kind of important, right? We have independent variables. And then we have dependent variables. The dependent variable is, this is the data that we're collecting. So the dependent variable in this case is in the first example would be the number of students that are awake. The second example is the number of days that the sun came up. The independent variable is more of like the treatment, like what are you doing? What are you giving? What factor are you changing here? In the first example, it would be the application of Red Bull. In the second example, it would be the crowing from the rooster. All right. This you have control over. As the organizer of the experiment, you control the independent variable. What you don't control is the dependent variable, the data that you're going to be collecting. This is the unknown, right? So just remember D for dependent and D for data, right? Again, when we make a hypothesis, we use what's called deductive reasoning, right? So we're creating a hypothesis to a certain situation or a certain experiment, and we're using this if then form of a statement. If I give the students Red Bull, then 100% of them will remain awake during the entire lesson. If I kill the rooster, then the sun will still continue to come up, or will not come up, really, because we predicted that the rooster is causing it. So if I kill the rooster, then the sun won't come up. All right. Now, when a hypothesis is tested, and in the conclusion, you're stating that the data is supporting the hypothesis. If this happens many, many, many times over, and is tested in many scientific communities by many scientists, we could graduate this hypothesis into what's called a theory. So once the hypothesis has survived rigorous testing without being disproven, then we refer to that as a theory, right? All right. So if it's supported by many lines of evidence, for example, evolution is a theory because it's been so tested and using multiple avenues of testing as well. And it's supported by many lines of evidence. I mean, specifically DNA evidence. It's kind of hard to refute. But, comparing embryos, comparing structures, things like that, analyzing fossils, you could see that species are changing. We could see it before our eyes, even with bacteria, that the bacteria are changing over time. This last slide says science is not perfect. Set in stone answer to questions about the natural world. Nope, it's not that. It is dynamic, right? Anything that we think we have proven today can possibly be disproven later on as new ideas emerge. So it's limited on what you can answer to using scientific exploration or scientific experiments. Like, what is the meaning of life? What happens after you die? Those are not the types of questions that science is designed to answer, right? Um, you know, generating a hypothesis and coming up with a controlled experiment. This is what scientists are trying to, answer. This is what science is designed to answer. All right, and that's the last slide. So what I recommend you do is take what we have here, right? Go back and read the book, and you'll see a lot of this information is very similar. And then look at the concepts. What's the scientific method? What are the steps? We kind of went over this. What is inductive and deductive reasoning? What's a variable? Well, we talked about independent and dependent variables. All right. What are the properties of life? What is homeostasis? Give me an example of negative feedback and positive feedback. We went over that. How are things organized? We looked at that as well. So this is a good place to look. These are great concepts to review. If you have any questions about any of the information that's in the notes or in the book, feel free to email me. In addition to these notes, we have to do our first lab, and that will come in a separate video. All right. All right. See you soon.